All right. Um, Good evening. I'm going to go ahead and get us started. Um, my name's Dave Powell. I'm the president at Wake Audubon. And I'm going to give us some announcements here at the beginning before we introduce our speakers for tonight. And so just bear with me as we go through the announcements. As many of you know, we have bird outings pretty much every weekend. If you're interested, you can find information for them on our meetup page. We have outings coming up this month at Durant Nature Preserve, Hilltop Needmore, Bass Lake, and others. We also have at the end of the month, on March 29th, we have a Let's Go Birding Together outing, which is focused for our LGBTQ plus and allies community. It's going to be at Horseshoe Farm Nature Preserve. If you are interested, you can find registration information on our meetup page. It will be 8:45 in the morning. If you want to arrive a little bit early, Rick and our other walk leader, Erla, will help make sure you get your binoculars all set up properly for that event. We also will begin doing our beginning birding workshops again this spring. There'll be information released on our webpage and meetup page soon. So keep an eye out for that. We have tons of volunteer opportunities coming up, so I'm going to try and work through those. We have two cleanups happening this month. One is doing a trash trap cleanup at the Walnut Creek wetland. This is in partnership with Sound Rivers, which was our speaker from last month. We were going to do it in February, but we had to reschedule. It's this coming Saturday on March 15th. And then we have an stream cleanup at Biltmore Hills Park. This is a cleanup we do like twice a year, each spring and fall. And that is coming up on March 22nd. So if you want to get involved in those cleanups, you can RSVP via our meetup page or find the information to be directed to there. All right. We have some other general volunteer activities or opportunities I want to talk about. So Wake Audubon is an all volunteer run organization, which means we're always looking for people to get involved and help us out with things. If you're interested in learning anything more about volunteer opportunities, particularly like leadership opportunities, come find me after the meeting. But we have positions on our different committees that we need help with. We have a Lights Out subcommittee chair we're looking for, a conservation committee chair, members of those committees and others. And we're also looking for a volunteer coordinator. So if you're interested in any of those things, let me know. We also have a special opportunity this year in that it's our 50th anniversary. And so we are planning a big get together for this fall. If you're interested in helping that out and helping out with that and shaping it, let us know. Either Luis or Anne, if they can raise their hands, find them, let them know you're interested in. We need help with event planning from little things to big things. So, and then we're starting to enter our spring tabling season. So we have two events that I know that we need help with table volunteers for. One is Ralston Blooms at the Arboretum. That's on April 26th. And one is Herb Fest in downtown Cary. That's May 4th. They're both in the middle of the day. If you are interested in them, you're going to want to get a hold of Alexa. We dropped her information in the chat. If you're in the room here, you can come find me and let me know, and I'll make sure you get connected. So those dates are April 26th and May 4th. One of our goals for this year is we're going to be starting a bird-friendly habitat certification program. It's just getting up off the ground. Our friends over in the New Hope Bird Alliance in Durham area run one. If you're interested in getting involved and you're in the room, come find me. Otherwise, you're going to want to email our acting conservation chair, Mary Abrams. And I also just wanted to point out that right now the city of Raleigh is going through a process to develop their next comprehensive plan. There'll be probably a variety of opportunities to be involved in shaping that. But there is an event next Tuesday, March 18th at Dix Park Chapel, where you can provide input and hear about plans for trees and urban ecosystems as part of that comprehensive plan. So if you're interested in that, you can find the information through the City of Raleigh, or it's in our newsletter, or check out our chat if you're online. Advocacy. So our Lights Out program starts soon on March 15th and runs through May 31st. During this time, we encourage people to turn off their outdoor lights after 11 p.m. So between 11 p.m. and 6 a.m., turn off your outdoor lights, close those drapes, down shield your lights if you can. That starts Saturday. We also have an information campaign to help spread the word about doing this to help with bird safety. So if you are interested in getting a sign to help spread the word, you can find Anne if you're in the room, or email her. We have a few signs left. So if you want one, jump on it tonight. We also have our Advocacy Day coming up. It is on April 29th. We do this in partnership with the North Carolina Audubon Society. It's at the state capitol. It's the 9th Advocacy Day. And if you're interested in doing that, you'll want to register. We'll drop the link into the chat for that. It's also in our newsletter, or you can find it on North Carolina Audubon's website. It's a great event. I might have a little bit of a bias because I'm a policy and political nerd, but I've gone the last 3 years, and it's great to get to talk with your representatives. And it's not as intimidating as it might sound. They're like totally normal people. So, who for the most part like birds and Audubon. So, all right. And then I also want to announce that our spring plant sale is starting. It begins again on Saturday, March 15th. It runs through the end of April. If you are part of Wake Audubon, you get a 10% discount, and there's a code for it, RC Woodpecker 2025. You will order through Field to Cottage, our partner in this plant sale, and you'll want to get on it because they do sell out of stuff. There's limited quantities. So this is a native plant sale, and a portion of the proceeds go to Wake Audubon. It's one of our main fundraisers each year. And then two more things. If you're with us for the first time this evening, let us know. We will pay for your membership in the Audubon Society for the first year. This includes membership in the National Audubon Society as well as in Wake Audubon Society. And so it's our way of welcoming new people here. So come find me if you're interested in that after the meeting. And then last but not least, I want to announce our next program meeting is going to be at a different time and a different location than normal. So in April, we're going to do our meeting on Saturday, April 5th at 1 p.m. It will be here at the museum, but it'll be in the Daily Theater. So that's in this building, but on the first floor? Yes, just to the left as you come in. And for that event, we're going to be doing, we have a storyteller who's joining us and is going to be telling stories that are bird related. She is a Grammy-nominated spoken word artist, so it's not to be missed. Join us at Saturday, April 5th at 1 p.m. Anything I missed, my fellow committee and board folks? Megan.
Make you aware that if you got an email from Wake Audubon today with a 1-minute volunteer survey to please fill it out. That information is helping us better understand how to be a better steward of volunteer opportunities to make sure you all are getting out what you hope to get from our organization. Like I said, it's 1 to 2 minutes max. Please fill it out.
Yes. All right. Anything else? Jeff?
We announced one of our members passed away yesterday. Bob Davis was a board member for several years and very active for many years, attending meetings and field trips. I think he helped lead some field trips. And he was a key team member of our Wildathon for about 20 years. We are still planning to do our Wildathon since Stephanie and me may be the only team members left, but we plan to do it this year, either late April or sometime in May. I don't think the date yet.
All right, thank you for letting us know, Jeff. I'll repeat those announcements. So Megan announced that we have a volunteer survey that went out via email today. We appreciate if people can fill it out so we can help know how to best present volunteer opportunities for you and match you to your individual interests. And then Jeff shared an announcement that one of our longtime members, former board member, supporter of Wildathon, passed away yesterday, Bob Davis. Is that correct, Jeff? Just want to make sure I get it right. Thank you for sharing that news, Jeff. I'm sorry to hear that.
Also one of my very best friends.
Oh. So everybody give Jeff a hug. That's his favorite thing after this meeting. All right. And
I would like to introduce my son, who will give a what you call it, elevator speech.
Sure.
About
Yeah.
field inclusive, and then I'll introduce our 2 speakers.
Sure. Awesome. Well, hello, everyone. Good evening. So my name is Lauren. I have been here before, but I have been absent because I've been neck deep in dissertation work. So I'm glad to be able to make it this evening. So hopefully, well, it will. Dissertation work will conclude in the fall. So I will definitely let you all know that, because my focal species is your bird of the year. So yay to the red cockaded woodpecker. But I was asked to say a few words about field inclusive. So if you all have heard about this organization, I'm sure you have, because we partner with Wake Audubon to give the beginning birding programs that we do. But Field Inclusive itself, just to give you a little bit of background, was founded by myself and my colleague while we were both PhD students, so at NC State University. Part of our research and part of our degree is going out and collecting data and doing field work. And so essentially, long story short, we noticed that there was a gap when it came to the support that was given to students and faculty and staff, whomever was going in the outdoors, when it comes to what we call social field safety. So essentially interactions with the general public. That often goes unmissed. When you're outdoors, people are questioning you, asking you what you're doing. It can lead to some very questionable, unsafe situations. So that's what we touch on, along with your basic physical field safety and everything like that. So we do a lot of work in that area. We have amazing partners. So Wake Audubon Society was actually our first ever partner that came on when we founded our organization in August of 2022. So I do want to give you all a round of applause there for believing in us. But tonight you're going to hear from 2 of the award recipients that your organization funded. So not only do we do workshops, advocacy work, various things like that, our beginning birders programs, but we also offer financial opportunities to marginalized and historically excluded individuals. So that's who our nonprofit really focuses on here. So anyone, that varies by race, religion, gender, LGBTQ, people with disabilities, neurodivergent individuals, everything under that umbrella. So 2 of those individuals you will hear from this evening, and these 2 individuals were the individuals that you all gave financial support to through our organization. So I'm excited to hear more about their research as well. So, but I'm happy to be here. Also, I just want to give a round of applause and shout out to my Raleigh volunteers. So Miranda, not to put you on the spot, but Miranda is here. Hold on, I'm just going to be nosy. So I'm going to have to yell at the rest of my volunteers because I told them to be online today. So I'm going to need to yell at them. But I do have other volunteers who hopefully will join later on. But also I want to give a huge shout out to Rick and to Phil, because without you both, honestly, this partnership probably wouldn't have gotten off the ground. So thank you. All right, I'll leave it at that.
Thank you, Lauren. Okay. Wake Audubon Society funded grants to 2 graduate students for their research in 2024, and Field Inclusive, as Lauren explained, administered those grants. These 2 graduate students will join us virtually tonight to review their research, which Wake Audubon Society has funded. I'm going to use my notes because these brilliant young women have done amazing things, and I don't want to forget anything. First up is My-Lan Lee. Sorry, My-Lan. She's a graduate student in the Integrative Biology Department at the University of Colorado, Denver. My-Lan has worked as a biologist and botanist in the Western United States since 2018. Her parents encouraged her love for wildlife early on. Her mother brought her to their local Santa Clara Valley Audubon Society chapter, and they volunteered to monitor Western bluebird nest boxes at their local wildlife rehab center. This past summer, she worked with other scientists to collect data and perform pollen supplementation experiments on the Avery Peak twinpod, a rare plant species that only lives in Colorado. Quantitative information about this species' ecology is limited. The research focused on measured ecological variation and reproduction to help fill knowledge gaps and better conserve this species in the future. After we welcome My-Lan, we'll have a question and answer period. Then I'll come back and introduce our second recipient. So welcome.
Thank you. I have to go ahead and share my screen, so hopefully that will work. Wait one moment. All right, I think do this. I think that looks right for me at least.
Yeah, it looks good.
Great. Okay, so yeah, today I'll be presenting some of my field research on a rare endemic alpine plant species. I know that I had told Anne, and it probably says in the announcement as well as on the website, if you were looking at Field Inclusive's website, that this species is called the Avery Peak twinpod. But I find that that name is a little bit overly specific because it doesn't, this plant, which is shown on the screen, doesn't only live on that one particular mountain. Like Anne said, I'm affiliated with the University of Colorado, Denver, but my advisor is actually at the Denver Botanic Gardens. And so I've been kind of working with these 2 different institutions for this research. I promise I'll talk about what some of these terms mean in a little bit more depth, but just a little bit about me, even though Anne talked about this already. Like growing up, I used to watch so many wildlife documentaries with my parents, but birds, I think themselves were my spark or catalyst for a love of nature. And as you guys can see, the presentation is about plants, but I do want to say that birds helped me find my spark plant because during college, when I was studying wildlife biology and I was in an ornithology class, we were asked to take detailed notes about the habitat and where these birds actually lived. And I realized that I couldn't talk about, this one shrub that I saw these bush tits interacting with. And some of the students who were in a field botany class that I would eventually take told me that the birds indeed we're in a California lilac or a buckbrush, which is shown in this photo, not my photo, this wonderful person, Kathleen Ryan's photo. But in any case, a little bit more about my background is that I grew up in the Bay Area. I was lucky enough to participate in the Western Bluebird Nest Box Monitoring Project run through the Santa Clara Valley Audubon Society during high school. And I learned a lot of my native birds through volunteering at a local wildlife rehabilitation center. I think that's what led me to pursue wildlife biology in college. I went to California Polytechnic State University in Central California, and through that program was able to work with a professor, Clinton Francis, who studied Western bluebirds, among other different species like lazuli bunting, spotted toadies, yellow warblers. I did an internship at one point where I was monitoring radiotelemetry for California condors, but it was really long distance telemetry, so I never saw them really up close. I still appreciated that. And after graduating, like Anne mentioned, I've been working as a technician and a crew leader, or sometimes also being classified as a biologist or a botanist in the West Coast, and also in the Intermountain West in Idaho, and now in Colorado. So I've worked with a lot of different study species, and I began my graduate program in 2023, studying this particular species, Physaria alpina. So just to break down some of those terms that I mentioned, specifically what really rare means in the context of this species. Obviously, rarity could be like an organism not being found in very many places around the world, or it could just mean that there are not very many of those organisms in the world. It also could be some combination of both. And in the case of my study species, its rarity has been determined by different scientists as being a G2 S2 rank. So for plants, in this case, it means that the G2 represents a plant being globally imperiled, and S2 means that it's also imperiled at the state level. My study species only lives in Colorado, and actually, despite that term of like imperilment, the populations where it does grow, even though they're limited, tend to be pretty big. Like if we're talking about something that's about to like blink out, there's a higher ranking or more, I don't know, like more of a dire ranking. That's like a G1, S1 for something that's critically imperiled. In any case, yeah, there's another description of some of that definition. And so, yeah, that 6 to 20 occurrences or populations of around 1,000 to 3,000 remaining individuals. Lots of plants and animals are in worse conditions than this, but this is about where we're at. And of course, that's different from like a rare bird, right? Where you're talking about something that's common in another place going to a place that it's not typically found. Okay. And as far as alpine habitats go, I know that, you know, some of you may have been in alpine places, visited, maybe like in the Appalachian Mountains, but generally I think of alpine as being above tree line, and the defining characteristics, it changed between different places. So in upland Colorado, alpine areas do not have trees and tend to be at very high elevations. And then that's a picture of one of my study sites. And, you know, I think that alpine habitats are like well known for these sort of like low growing plants. They can be quite beautiful in the summertime. While it's almost desert-like in nature, it's still quite beautiful. And perennial alpine plants that grow year after year are often very slow growing and quite sensitive to disturbance. So can be easily affected by things stepping on them constantly, and potential changes in the weather. Even though they're rather tough, they can handle like these long and cold winters, are often buried in the snow for most of the year. And just another visualization of what that means. You can see that like if we're going up a mountain, the alpine tundra, which is the only place that my study species really lives, only begins at around 11,500 feet. In that image, you can see that there's little sorts of half kind of looking trees around 11,000 feet. And those are meant to represent those sort of like windswept conifers that you start to see at really high elevations. And in Colorado itself, we don't necessarily have a ton of alpine, right? I mean, more so than other places, but just to give you an idea of how much land mass that really looks like. The white color in this map of the state represents all of the alpine areas of Colorado. But then for my species, it's only really found in a few spots of those mountain tops. And so those areas are called the Mosquito Range and the West Elk Mountains. And that brings me to another term that was in my title, which is this endemism, which means that these species are only found in this particular area. And I just loosely am defining that as the Southern Rocky Mountains of Colorado. And then this is, just if you want to see where these locations sort of fall in respect to other parts of Colorado. We're on the western slope, so we're definitely southwest of Denver in particular. And then if any of you are familiar with like Aspen, while it's not in that area, you can see that some of the points are around those mountains. And just a little bit more about my study species. I think it's pretty, but I'm biased. Again, it's also called that Avery Peak twinpod, which is a name that it got from the mountain where it was first described. It's a type of mustard, which you can recognize based on the fact that it has 4 petals for each of those little flowers that it has, as well as some other traits. It is a perennial species, which means that there's like an unknown lifespan. Well, not that that perennial means unknown, but there's not that much research done on a lot of rare species, so, and especially on plants, and so we don't really know how long they can live. And then below, there's a little picture of its fruit, which is why they're called twin pods. We have other Physaria species in the US, and they're all kind of called twin pods because their fruits inflate like that and have those 2 nice chambers. I think that's also pretty cute. So there's also a seed bank collection of these plants at the Denver Botanic Gardens for future research purposes, and just fairly limited research overall. Like these plants were described and that's kind of about it. There's 2 other sets of researchers who have begun doing research on this species in the last season and few years. And so I've kind of worked with them just to make sure that my own research wasn't really like overlapping too much with theirs, so we can kind of all focus on different things. But the reason why I was interested in studying this species in particular, despite it being imperiled and not very much information being known about it, is that one of the other researchers who had made that seed bank collection noticed that there were some pretty big differences between 2 populations that she visited in order to make these collections. One of the populations, sorry, let me make sure this is coming through. She noticed that the plants were making quite a lot of fruit, and so that's represented by those little popcorn looking things, the twin pods. And then another population she had seen flowering earlier in the season didn't end up going into fruit. So there was obviously something different happening at these populations that might be affecting their reproduction, but we just weren't sure what those variables might be. And because the nature of those rare plant rankings is determined based on the condition of each population, and we just couldn't gauge whether that was normal or not for the populations to have such different amounts of reproduction, I just wanted to investigate that further. So rare endemic species that only live in particular places for whatever reason, right? Whether it's that that habitat is the only habitat they can do well in, can be affected by environmental change, right? And we're missing a lot of the ecological information about these species. And so I wanted to know, like what I said, what was the ecological variation found in these populations, and what might cause those differences in reproduction. And we can hypothetically think about a lot of different studied reasons for reproduction being different. And so in this diagram, I have my study species while it's flowering, so like during its reproductive phase when it's receiving pollen and potentially like making fruit. And so in these 2 scenarios, hypothetical scenarios, we have a situation on the left where the plants are experiencing some kind of condition that makes them make more fruit, and then on the right, some kind of set of conditions that make them make less fruit. So based on the literature, some of the ideas that could lead to this difference include potentially abiotic factors, right? Maybe one site's getting a lot more water, maybe there's more like light exposure, who knows, right? But based on what I saw, it didn't seem like, at least on a very broad level at the sites, that they were very different. That's one thing that could cause some kind of difference, right? Another difference could be same species density. So I was imagining that maybe more of the same species of our plant being in the same plot or area might cause a higher level of reproduction, because maybe with this kind of increased floral display, the population would be more attractive to pollinators, right? The pollinators might come and visit and thus pollinate more of the flowers, resulting in more fruit. There could also be a situation where if there were lots of other kinds of plants in the area, the pollinators could be distracted by those other species, might be choosing to visit the other species more often. That could be something that could lead to less fruit potentially. And so that's maybe that idea of competition, specifically against like other species. And then there could also just be a situation where there are no pollinators at a site at all, leading to differences in reproduction, right? I can tell you more about how I collected the data, but I decided I would just put at least one of my preliminary research results here, which is that I'll try to break this down, but basically what I found is that this was opposite of what I thought about the same species density, and that the likelihood of an alpine twin pod going to fruit was more strongly predicted by lower numbers of its own species in the plot that were flowering. So there was this kind of negative correlation with the number of same species neighbors for a plant in a small area and how much fruit they made. And so that's a signal that there's some of this intra-specific competition, so competition within the species. But exactly why that is, whether it's something about the floral display itself or the plants themselves are taking up like a lot of water or other resources, we don't know quite yet. That was basically, this result came out more strongly than any of the other factors when I factored in the cover or presence of other kinds of flowering species. So that was a bit interesting. And so measuring and comparing these ecological conditions and this variation will hopefully address some of those knowledge gaps for those rare species and might be beneficial for future research and continued management, right? So just to get into a little bit quickly about how this research was set up, when I went out to these sites, I would go and like use this little quadrat to determine how many other individuals were in an area, and that was standardized across different sites. You also see that there's this little logger here, that white kind of post on the left-hand corner of the plot that is a data logger. And so this device, and there were many of them that were deployed at different plots, was picking up temperature and also underground moisture at that specific site. So we have some really hyper-specific abiotic data that is associated with a lot of these plots. You might also be able to tell, I don't know how big your guys' screen is, that there's a little QR code kind of on top because of the data logger, and that's a sunscreen or sun protector. And often people visiting these sites like wouldn't know what the data loggers were. And one of the other researchers had heard someone like leave a note on top of the sunscreen asking what it was. And so this season, I put up these little QR codes, which directed people to a small message and also an email so they could ask me more about it, especially because, you know, concerned, I don't know, like hikers and people who are recreating might think that these devices, they might just not know what the devices are. And so we have a photo also of one of my undergraduate field assistants counting the number of twin ponds in the area, and probably at some point she's later after that she would have taken like the percent cover, assessing kind of how much of those plants were actually in that plot. But in order to even implement the research at all, I had to first identify feasible research sites, apply for and obtain permits, design a protocol to answer those questions, and figure out our data collection. A lot of that was facilitated with help through the Denver Botanic Gardens. I also then went ahead and recruited and trained undergraduate students in these data collection methods. And because this is research done on a rare plant species, I'm going to eventually share my final results with the Colorado Natural Heritage Program, which tends to monitor rare plants, the Colorado Native Plant Society, and the U.S. Forest Service, where all of this research was taking place. These are some of my undergraduate field assistants helping me with the various aspects of my research. I definitely couldn't have done this without them. And in that vein too, the support from the Audubon Society was really essential for me to make sure that they were having a safe and kind of beneficial experience. Sorry, that all came up at once, but as far as awards go, obviously the Wake Audubon Society and Fields Inclusive helped me, but I also had some help from the Colorado Native Plant Society and the Garden Club of America. And with all of these awards together, I was able to achieve a lot of things this summer that I would not have been able to do otherwise. The thing that I really feel that was the most important was that I was able to use my support to fund my undergraduate's wilderness first aid trainings. In field work, I don't think that you can possibly overstate the importance of field safety. I know it can like seem really fun, like we're all just kind of like running around outside looking at plants, but most places, you know, like have a lot of potential risks. No matter where you are, you could experience like heat exhaustion, hypothermia, at least at like my sites, all of these things that are not actually very common knowledge. And I think it's really important that students have the training for that. But I didn't necessarily have the funds to do that with my own kind of research support through the university and the gardens. And so this funding allowed me to cover that for 6 students as opposed to just 1. I also was able to cover my camping and lodging fees and a subscription for satellite phone use. So when we were out of service on our like multi-day camping trips, were able to still check in with you know, my advisor so they knew that we were okay. And if we got into trouble, which thankfully we never did, we would have a way to actually contact someone. And I also use the funding to help supply safety gear and research supplies like those data loggers, which I think will be really helpful in the end. It'll take a lot longer to analyze that data, and I don't anticipate to really work on that until this summer. But it's seeming like from kind of the broader data measurements that I took that there isn't like that maybe some of the answers about reproduction will be found in the microhabitat. So specifically those smaller measurements made that are specific to each of those plots. Something else that I was able to do that I was glad for, especially with all of my undergraduate students' help, was to document pollinators, herbivores, and parasites of this particular species. I don't think anyone else has done this formally from what I've found. And this is just a snapshot of things I've logged in a citizen science database called iNaturalist. Many of you are probably already familiar with it. But I'm looking forward to trying to summarize these findings to make a natural history note about them. There was like one particular herbivore that was just all over alpine mustard plants and I was not sure what it was. It was everywhere, and I just couldn't find anything about this little grub that was eating all of the alpine mustard species. But after consulting a bunch of entomologists online, it's most likely that these are red turnip beetles, and they haven't been formally documented as predators of Physaria Dreba ericimum, which are all other kinds of mustard genera. This summer, I think I'm going to apply for a permit so that I can go and collect some of these grubs and rear them in a incubator in order to confirm that that's what they actually were, because I just saw a bunch of them at the beginning of the summer, and then I came back later and then saw some of these red beetles, but it's not entirely sure that, that's what they became. And just because I thought it's fun to see what other rare plants lived at some of these study sites, these are some other examples of plant diversity too that could be found where the alpine twinpod lives. We have some like Ipomopsis and paintbrushes and these sawworts, as well as these tansy asters, beard tongues, and milk vetches. I just always like an opportunity to show a picture of a pretty plant. In any case, I hope I'm not too much over time, but thanks so much for your time today. And I'll happy to take any questions. And yeah, I can leave this up, but I also could stop screen sharing. Let me, yeah, I'm just gonna, maybe I'll just leave it here for now, I guess, and then I'll stop sharing when we're done with the Q&A.
Well, thank you so much. What an interesting talk. Thank you. Are there any questions here in the room or online for My-Lan?
I'm checking online. I'll just start with these first.
Okay, don't worry, we'll pass them along to you. Megan and David are going to find them online for you. So you can just focus on answering. There was a question right here. I'm sorry. What was your question? Okay.
Hi, My-Lan. That was a terrific presentation. I really enjoyed it. I wanted to ask about, sort of get back to your, what you were thinking when you chose this plant to focus on. Was it a particular interest in preserving this plant and trying to find out more about it since there's not much known about it? Or are you using this more as a sort of an example species to develop a broader and exploring approaches to develop a broader strategy for conserving rare plants? I'm not sure that makes any sense. But.
Could you hear that okay?
Yeah, I think so. And if I don't address the full question again, please just let me know. The question was asking about kind of the development of this research question, specifically around the study species, and whether I was more focused on the study species itself, and that or using my experiences to develop a kind of protocol for plant conservation. And I think there was a little bit of both things. Having that kind of spark of seeing those two populations reproduce really differently in previous years made me want to study it, but also because there's already some other researchers studying this species, I thought it would be helpful to try to address something that they weren't covering, which was making comparisons between populations. And then with those data loggers in particular, I'm hoping that my work with them can help the other researchers I know who have begun to utilize them as well, use those as a part of our research on plant species in the future, just because I think it'll take some time to figure out the best way to analyze that data. And that part, collecting it's one thing, but analyzing it's another. And in my work, I'm hoping that kind of my eventual products will create that workflow that you were talking about that could help other researchers.
Thank you. That did answer her question. Thank you. Is there any other questions here? Okay, Rick has a question. One moment. Okay, we're handing him a microphone.
It's not so much a question. I just want to commend you on the way that you introduced your topic. It was so relatable, I found. And I love the fact that you laid out all the hypotheses and then the finding was sort of counterintuitive to one of your hypotheses. It was like this big aha moment. And so I just want to congratulate you on a really wonderful presentation. Thank you.
Thank you. How
Thanks so
about,
much.
oh, excuse me, how
Sorry.
about online, Megan, or Dan?
Can you repeat the question, please?
Can you bring the microphone to her, or? Okay, here comes Megan. She has some questions.
Cool.
She's observing, and who were the pollinators or the wasps in the photos? So how big were those plots? Who were the wasps?
Yeah, I'm also able to see that question online. So the plots, oh, sorry.
Yeah, that's good.
Go ahead.
Cool. The plots were 1 meter by 1 meter, and so I visited. There were 3 sites with 20 plots that were visited twice, and so that's kind of the information I was addressing in my hypotheses. But I also visited another 3 sites that had a combined total of 50 plots. So in total, like 110 plots were visited at least once. And then the pollinators themselves, like what we're seeing on this last slide here is actually this is some kind of fly. It's like called like a micro bee fly, which is a very confusing name, but that's a kind of fly. But it just seemed like these plants were getting a lot of visitation across a lot of different groups, like bees, flies, beetles. I caught at one point like a mite or of some kind in one of the flowers. I also saw a lot of ants visiting the plots. So it doesn't really seem like at least I can't make a conclusion of exactly whether or not the pollinators who were visiting were resulting in increased seed production, because sometimes, insects or other arthropods visit, or even, other animals visit flowers and it doesn't result in successful fruit production. But as far as attracting different insects and arthropods, these guys just seemed really popular with all kinds of critters.
That's good. One more question, I think, and then we'll move on to Cheyenne Reuban-Thomas. Are there any other questions online or here? Did you have bear safety training? That's the last question.
Yeah, I think what's really interesting about wilderness first aid trainings is they tend, even though they're like official certifications, they tend to vary depending on the places that use them. And I tried to make sure all my students went through a training that was run through like a Coloradan program. So I think because we only have black bears in the Rocky, at least the Southern Rocky Mountains, they just talked briefly about like best practices, like not, leaving food out and making a lot of noise when you're moving through an open area. But just generally given these alpine environments, we weren't really seeing them, or I never see black bears at the actual study site. But because we travel through black bear habitat, like going up the mountain through subalpine and montane forests, I think it was good that we covered that anyways. Just good for anyone who's recreating outdoors where bears live.
Yes, thank you again, and we'll say goodbye to you, and thank you. Go ahead and stop.
Well, I'm going to stop sharing now.
I would like to introduce. Okay, now I would like to introduce the second grantee, Cheyenne Reuban-Thomas, who is a PhD candidate in Ecology and Evolutionary Biology at Cornell University. Cheyenne is comparing the impacts of traditional ecological knowledge, TEK, practice systems versus settler land management strategies on insect biodiversity. In other words, she's comparing the impact of cultural fire as TEK and prescribed burning as settler management practices. She collaborates with the Oneida Nation Bird Monitoring Program, which is part of the a collective with the Great Lakes Audubon Society. She also collaborates with Indigenous communities whose lands she works on to connect with those community members in deeper ways. She is inspired by the strong Indigenous women leading traditional fire practices for their communities. Cheyenne's research will help fill gaps in our collective knowledge of the impacts of controlled burning on insect biodiversity. Her field work occurs in two places: remote oak woodlands in the Yurok territory of Northern California and the prairies of northeastern Wisconsin on the Oneida Nation, which is her home nation. Please welcome Cheyenne Reuban-Thomas.
Okay. Hi, everyone. Sorry about that. I was on mute. I just want to make sure that everyone can hear me. You can see my screen okay. Okay. Okay, cool. All right. Sagolis Kweku, Ganyantewan, Niogato Niantawaka, Wankanto. So hi everyone. My name is Cheyenne Reuban-Thomas. My Oneida name is Ganyantewan. I'm an enrolled member of the Oneida Nation of Wisconsin and of the Tonawanda Seneca Nation. I am also a second-year PhD student at Cornell University in the Department of Ecology and Evolutionary Biology. I really liked My-Lan's introduction slide, but I really didn't prepare one, so I just want to add a little bit of background. I've been a lifelong learner of the land. I'm lucky enough to know my culture and its values and teachings, and having always been interested in ecology and biology, I got my bachelor's degree in conservation biology at Buffalo State University. And engaged in research focused on population genetics of eastern hellbenders, which is a culturally significant species in my Seneca community. I then worked at the Department of Agriculture for an invasive insect program as a tribal liaison to ensure that traditional methods of species eradication were respected and upheld by the government. So I knew I wanted to go back to school after my love for insects grew through this work. So I ended up getting my master's in ecology, evolution, genetics, and genomics at the University of Rochester. And there I studied the range expansion of a social wasp. So despite that, I was still missing the values and teachings of my community, which is how I ended up where I am now. So this presentation will review my ongoing doctoral research. It's still very much ongoing. And so my goal really is to compare the impacts of traditional ecological knowledge and settler colonialism on insect diversity. So we'll jump right in. All right. So we may have all heard this general statistic when reading literature about biodiversity and Indigenous peoples. So in 2008, Soberville stated that Indigenous peoples account for less than 5% of the world's population, but they steward or manage about 22% of global land territories. And with that, about 80% of the world's biodiversity is sustained on these Indigenous lands. And though this statistic is difficult to empirically determine, there is actually still evidence of increased biodiversity on Indigenous lands when compared to those lands completely occupied and managed by settlers. So focusing on what is now called the United States, Indigenous peoples and communities across Turtle Island have lost close to 99% of their ancestral territories from settler colonialism. And this statistic is reflected in the distribution of land management authority today. So the federal government manages 640 million acres of land across the United States. Meanwhile, federally recognized tribes hold rights to only 56.2 million acres. Even today, we still see the impacts of settler colonialism and Indigenous land dispossession through loss of inherent rights to stewardship of ancestral lands. And despite this loss of land, observed biodiversity is often higher on lands with Indigenous-led land management. So a leading example of this is actually the Oneida Nation of Wisconsin, my home nation, who is buying back land and restoring habitats such as prairies and forests to their natural conditions. So So the UNOTA monitoring programs have observed that many animals are actually returning to these lands, including species of concern across different taxa, such as reptiles and amphibians, small mammals, and birds. And birds actually play a large role in shaping fire regimes on the United Nations reservation, which I'll talk about a little bit later. So these programs really support animals that are having trouble thriving outside of the reservation, including those that are considered either endangered or threatened in the state of Wisconsin because of habitat loss as a consequence of settler colonialism. So traditional ecological knowledge, which I'll now refer to as TEK, is acquired generational knowledge gained through oral tradition, observations, and practices. And it's likely a main factor for sustaining these concentrations of biodiversity. And many forms of TEK exist and are practiced to this day, including increased popularity outside of reservations and Indigenous communities. So just two examples of this include sustainable agricultural practices, such as companion planting of the Three Sisters, and this enhances soil fertility. And then we have traditional fire practices, which are referred to as cultural fire. And this is utilized to restore habitat on tribal lands by promoting growth of native plants or medicines. And with this in mind, most efforts to protect and conserve biodiversity are focused on culturally significant species. So this graph shows taxonomic distribution of those recorded across the world, and plants are by far the most sustained group, which are broadly significant for Indigenous agriculture, medicines, and art. And following this trend of favored taxonomic groups, there's advocacy for protection and restoration of culturally significant mammals, which have resulted in successful restoration programs, such as mammals, including bison for Haudenosaunee nations like Oneida, fish such as coho or Chinook salmon, which are significant to the Yurok, who are actively working to restore these populations in the Klamath River, and many bird species. There's been pretty successful restoration of California condor populations with the Yurok Condor Restoration Program. However, insects are often overlooked in conservation efforts, despite their pretty significant role in maintaining healthy ecosystems. And even among Indigenous communities, including my own, who don't happen to hold much cultural value for insects, they tend to be neglected in these contexts. And as many of us know, this lack of attention is unfortunate because of the alarming decline in biodiversity of insects worldwide. So from this paper, Wagner et al. Describes the many factors contributing to these losses in insect biodiversity, all of which are pretty much driven by settler colonialism. So factors such as urbanization, habitat fragmentation, and Western agricultural practices like monoculture, they can all consequently influence patterns of this biodiversity loss that we see. And recently, there's been more interest in how one of the most abundant insect families in the world responds to anthropogenic factors. So we're talking about the ants. So in this paper, it's emphasized that ants can potentially serve as bioindicators of environmental health. And studying responses to these, or of these ants to these factors, has the potential to actually inform strategies in biodiversity conservation. So ants are my ideal model system. Ants are an ecologically beneficial insect family, fulfilling many roles which are significant for environmental health and sustainability. And the ant family consists of more than 14,000 described species, and they are found almost everywhere. So they occupy diverse ecological niches. They're also studied extensively in ecological and evolutionary contexts, which allows me to investigate longer-term impacts that TEK may have on our insect kin. And so I'm just going to backtrack just a little bit because I have to talk about fire. So because I'm interested in investigating the impacts of TEK on insect populations, we just have to focus on, or discuss my focus on cultural fire as a TEK method, which Indigenous communities have utilized for millennia. So the practice of fire over many generations has promoted fire-adapted and fire-dependent ecosystems. And here we see an example of maintaining an ideal ecocultural site on the Yurok Reservation. So this is an ideal ecocultural site because it's on a burn schedule that allows for straight growth and ideal pliability or flexibility, which is favored by elders and community members for basket making. And as mentioned earlier, cultural fire can also be used in support of habitat restoration and food sovereignty efforts. So most of us are familiar with prescribed burning. Government agencies have realized the importance of wildfire fuel reduction as an additional benefit to cultural fire, and they have since adapted this use of fire as a land management strategy. However, it's important to note that agency-led prescribed burns differ fundamentally from cultural fire. There's more connection and intention behind cultural fire, specifically considering end goals for these burns. And cultural fire is rooted in Indigenous knowledge, of course, and TEK practice systems, which consider relationships to our more than human relatives. Agency-led burns lack this generational knowledge and use of traditional practices or methods, often burning outside of culturally defined windows and even burning too hot. So I'm really interested in the broader question, does use of cultural fire increase insect diversity? In other words, what are insect kin telling us about the differences between Indigenous-led and agency-led burns? And so to investigate this question, I'm in collaboration with Indigenous communities who have revitalized their uses of fire following US fire suppression. And so I'll refer to these as cultural fire sites. On the West Coast, I'm working with the Yurok tribe of Northern California, who have fully reclaimed their rights to fire about 20 years ago. And in the Midwest, I'm working with my home nation, the Oneida Nation of Wisconsin, and they've revitalized their fire practices within the last 10 years. And so each cultural fire site is paired with an agency site of similar landscape structure, including similar elevation and climatic factors. These sites are, however, managed by the government, of course, but they do match habitat restoration efforts to the cultural fire sites. So in California, I'm working with the US Forest Service, and in Wisconsin, I'm working with the State Department of Natural Resources. Federal and state agencies undergo the same training, methods, and permitting requirements for burns, so it equals out in the end. And as personal communications and observations from this past field season, agencies do burn out of culturally defined windows, and their severity actually may be too intense. In other words, it looks like they burn too hot and too quickly. So these might be large factors in their expected differences in impacting insect biodiversity. And so the pairs of field sites are pretty distinct in that California sites are oak woodlands and Wisconsin sites are prairies. There will be direct comparisons for each sites or for each state or landscape, not between states. This will allow me to determine if patterns of biodiversity are consistent across different landscapes, but also different regions. And to address this broader research question, I'll be asking two things. So does cultural fire increase species diversity of the ants? And does it increase genetic variation of the ants when compared to agency sites? And so I completed my first field season this past summer. I'm in the midst of sorting and identifying all of my insect samples from my fieldwork. So I am assessing overall terrestrial insect biodiversity, but I'll be focusing on two prevalent ant species to zoom further into that diversity through species diversity and genetic analyses. And so just for the purposes of this presentation, since those species have yet to be determined, this could potentially be species from the genera Lasius and Formica, both of which comprise mostly like ground-dwelling species and generalist foragers. And so generally good fire, and when I say good fire, I really mean like controlled burns of any kind. Good fire should help restore and maintain healthy soils by replenishing nutrients, nitrogen and carbon. And so healthier soils provide favorable habitat for ant nesting and foraging behaviors. And because these behaviors generally increase success of ant colonies, but agency-led fire may be too severe, I predict that cultural fire actually results in greater gene flow and therefore increases genetic diversity within a colony because of the care and intention that goes into traditional or cultural fire applications, especially when comparing to those government-led prescribed burns. And so first I'll be analyzing ant species diversity. And so I'll be looking at the presence absence data and species richness in the chosen ant species. So I'm also going to calculate alpha and beta diversity using the Shannon and Jacquard indices. And these are just diversity metrics, ultimately informing us if or what the species richness is at each site and if paired field sites are more diverse than one another, respectively. And so for this, I'm expecting to see higher ant species diversity and richness at cultural fire sites than at agency sites. So I'm also assessing ant genetic diversity. This is for any of the geneticists out there, I guess, more so, but I'll be calculating population genetic summary statistics, such as nucleotide diversity or pi and Tajima's D. So these can tell me how genetically diverse populations are from one another, as well as show evidence of positive or negative selection throughout evolutionary history. And for this, I expect to see higher genetic diversity and positive selection or possible population growth at cultural fire sites as a response to successful adaptation to cultural fire. And so through this research, I hope to highlight the importance of Indigenous-led conservation and land management strategies by investigating the impacts of TEK practices on our insect kin. So this work will not only enhance our understanding of the benefits of Indigenous-led conservation strategies, but also hopefully restore environmental decision and policymaking power to Indigenous communities who have these ancestral rights to these lands. So in other words, that's really what we mean when we say land back as original stewards of the land. Now I want to take a moment here to talk about the Oneida Bird Monitoring Program that I mentioned earlier. I'm lucky enough to partner with them through my own work. They've actually been out in the field with me as well. So this program is a partnership between the Oneida Nation of Wisconsin, the University of Wisconsin-Green Bay, the Northeastern Wisconsin Bird Alliance, and the Audubon Great Lakes Society. So it's a volunteer bird monitoring program that focuses on bird surveys at restoration sites on the Oneida Nation Reservation. So surveys occur in various habitats, including marshes, forests, and you guessed it, prairies or grasslands. And breeding and migratory surveys are conducted throughout the year. So from these surveys, they actually now have records of state threatened birds, such as Henslow sparrows and grasshopper sparrows. So the goal of this program is really to monitor the biodiversity on the reservation, help inform land management decisions, and weave Oneida culture and language for community outreach and education. So prairies are threatened ecosystems which have historically evolved with fire, and Oneida is restoring their wetland and prairie habitats with prescribed burns. So the survey data provides insight into what management works best for bird diversity and prairie habitats, which might look like rotational burning, rotational grazing, occasional mowing. Ultimately, they're working toward bird-informed land management. And while my research does not directly involve them, there's a lot to consider regarding impact on birds, other than its obvious connection to adaptation or dependency on fire regimes for habitat restoration. Take that Henslow sparrow, for example. They are most happy in these habitats with those tall grasses and scattered shrubs, much like prairies. And their nests are usually well hidden on the ground in the midst of these tall grasses. And so they also do most of their foraging on the ground, which includes mostly seeds and insects. So Henslow sparrow diets include crickets, grasshoppers, beetles, etc. And while I'm focusing on ant diversity, I'm also assessing overall terrestrial insect biodiversity. So since I've sampled anything that fell into these standardized pitfall traps, of which include many of these insects, so essentially, if cultural fire sites where Henslow sparrows have been found do have greater insect biodiversity, there's evidence for support across taxa by these restoration efforts. And so I'm not done yet, but I want to acknowledge my advisor, Dr. Corey Moreau, and the Moreau Lab, who've provided insightful feedback on the work, as well as the communities that I'm working with the collaboration and knowledge sharing from each community. It's so insightful and meaningful to this work. Obviously, I'd like to thank the Wake Audubon Society. I received this research grant from them through Field Inclusive, which helped support my fieldwork this past summer. So it helped me buy necessary fieldwork supplies for sampling insects, as well as topping off a field assistant stipend. Personally, I grew up without access to some pretty fundamental resources and lacked access to opportunities like fieldwork, especially in college. So really, I'm passionate about kind of dismantling these barriers that folks from underserved communities face in STEM fields, because I know exactly what that's like. And so part of that was wanting to provide a livable stipend to an Indigenous college student. And I understand that hitting pause in life to take part in this kind of work that someone may be passionate about or curious about is not always possible. So I really wanted to lighten the load for someone interested in doing this work, which is exactly what I was able to do. And so one way I also help dismantle these barriers in my work that I always like to mention is my work as a junior faculty with a summer internship for Indigenous Peoples and Genomics of Canada program. I'm really passionate, again, about that community outreach and education. So I get that filled by teaching through Indigenous data sovereignty workshops and even local outreach with the Seneca Park Zoo in Rochester, New York. And but through SING, we assert sovereignty and governance in research and our data through training and supporting Indigenous communities wishing to lead their own research. So really we focus on training the next generation of Indigenous scientists who can share knowledge and resources. Like we have mobile sequencers and we share those with their communities as well. But that's something I always like to mention. I hope I didn't go too over time, but I want to thank you all again, Yonko, for listening to me. And yeah, I'll take any questions.
Yeah. Well, I found a way.
Okay. Thank you, Cheyenne Reuban-Thomas. Thank you so much. Are there any questions in the room or online? Okay. How about, I guess Megan is gone. Did you see any questions online, Dave? Oh gosh, you blew them away. Oh, we do have one question. Okay.
Great presentation. I don't know if you can hear me well,
But not here. Okay.
I guess I'll just repeat the question. So basically, in your talk, you talked about the differences between the two types of fires, one led by Indigenous people and one led by the federal government. Was there any effort in trying to quantify the differences of the fire in terms of maybe how hot they got, the carbon balance after in the soil, or the nitrogen balance as well? Was there any kind of abiotic factors that you tracked before and after the fire to kind of give you a concrete difference between the two methods? Because you talked a lot about how you're planning to distinguish the genetic variations between insects, but a lot of that is kind of driven by these abiotic factors. So your argument is kind of you're inferring that there's a difference in these two methods, but was there any kind of effort into actually quantifying and kind of giving the differences between those two methods?
Could you understand that, Cheyenne? Okay.
Yeah, I actually got that. Thank you. Yeah, that's actually a really good question. And it's honestly something that I didn't think was going to be so stark until I actually went to the field. You know, especially Yurok's a great example. Yurok, they have lush understory with their hazel understory, and even 3 months after the burn, and their fire scars, their tree scars are only maybe 10 feet high, whereas the Forest Service site that I visited, 3 months after, same timeline, it was barren underneath, and the fire scars went up like almost 40 feet. So that's indicative of too high severity. But unfortunately, the technology for quantifying the severity of fires isn't, I mean, it's there, but it's very scarce. I don't really have the funding for something like that, but looking into, you know, the soil composition is something that I am considering for the future. This is something I'll have to revisit my field sites throughout my PhD every summer for the next 4 years or so. So that is definitely something that I want to incorporate. I'm open to any ideas if you have any. But right now, it is kind of up in the air as far as that goes.
You know, related to that, I'll comment. I do a bird project on a site that gets burned every few years. We had some students come down from a local university to try to look at the intensity of the fire. So they have some kind of device they can put on a pole that's elevated, and it goes up 15, 20 feet and takes all these measurements. A few years ago, the fire burned really hot, and the flames went up 20, 30 feet, and it destroyed their very expensive equipment. And they haven't been back since. It was thousands of dollars of a special sensor on a pole. They said they'd never seen a fire get that hot. And so, yeah, it can be really expensive.
Yeah, I've heard similar stories with drone technology too. So that might be out of the question for me as a little PhD student.
Are there any other questions?
Just about my ants, because with all the species of ants, it seems like right up to 45 species of one type of ant. Why not choose an insect with fewer species or subspecies? I guess it just seems like that's.
He's wondering.
Yeah.
He's wondering why ants. There are so many species of ants. Would you choose an insect with fewer species?
That's such a funny question. I actually kind of just fell into ants. If it were up to me, I would probably focus on true bugs. But ants, my advisor, she's one of the leading ant people in the world, really. So it's kind of it is my choice, but it's not my choice at the same time. But there are options too to look into beetle diversity, because it is quite stark in Northern California. So that might be a possibility too. But that might be my undergrad. I have some undergrads doing some of my identifying, so they might do little side projects focusing on something else.
That sounds good. Okay, one more question if there are any more. Okay, here, Diane. Okay, one moment, Diane. Could you speak into the mic? She has a comment.
So impressed by these young women getting, or men, whatever, getting grants from us, Wake Audubon. That's another reason to contribute to Wake Audubon for the good things that they're doing.
Yes.
Thank you.
We think so.
Yes. Well, thank you again, Cheyenne Reuban-Thomas.
Thank you so much for having me.
We'll give a goodbye to you. Okay, or do you have another question, Susan?
Make this opportunity available.
Out to Field Inclusive. Yes, thank you, Field Inclusive. Okay, thank you. I'll let you all get back to your field work and, and reading. Thank you
Thank
again for
you,
joining
thank you.
us.
Thank you for having us.
Yes,
Yes.
thank you so much. This was a really lovely evening.
You're welcome.
I just want to close out real quick by saying that about a year ago, we approved to provide more research grants through.
Oh, okay, I guess. They tried. Oh, well, okay.
All right, well, good night. It was
Yeah.
great hearing more about your research.
Yeah, no, likewise. That's like, I feel like I should have known that you already were working on with like kind of like Yurok area data as well, but just given like where I grew up and stuff, I know so many people went to Cal Poly Humboldt and or who are still there. And yeah, like the fire alliances and stuff are very strong, I feel like, in those areas. Like prescribed burning training and things that like try to think about those things. But yeah, definitely like I've been on a burn and I was like scared the whole time, and I felt like and like I remember the other firefighters saying like, oh, it's going to like it feel like it's going a little slowly because like we are here with the state and they've never done it, and they're like very, and the Cal Fire who was with us wasn't used to that. And
Yeah,
so they were going really slow, but then I was like, shouldn't it be slow? It's like scary, you know what
it
I
should,
mean?
right?
I was
Actually,
like,
they don't even have to
yeah,
have Cal Fire on their fires anymore. So
yeah,
completely independent now, which is great. So
yeah,
we can do it their way
that
without
is
getting any
cool.
flack.
Yeah. But anyways, yeah, this was nice again. And I assume that since we haven't had Sydney or anyone else come back, that we should be okay to go.
True. True.
But yeah. Okay.
Well, thank you guys. Thank you guys for having us
Thanks
again.
for those of you who are
Have a good night.
All right, bye.